So I'm just going to be covering basically all the information that they covered this week. Like I was saying earlier, it's all about diseases. However, these like first couple or so, they're about like different stages of meiosis that you guys haven't really talked to too much detail about before. So pretty much all of those are fair game. It's kind of like sub stages of meiosis is kind of the way I think about it, especially like prophase. Um, they break that one down into I think like 4 or 5 different ones. So those are all kind of questions they could ask. So I included this slide here, and I put it in the notes to know the different levels of organization. So what I mean by that with DNA packaging is I mean like it starts off with just DNA right by itself. So that's like this up here, right? Of course. So just DNA double helix here. Then they have kind of like the beads on a string type deal, which is he doesn't even cover that in text, so I wouldn't even think you'd have to know that. But once it kind of packs with histones, so DNA, if you notice from this picture, they wrap around histone molecules. Now it goes a little bit more into detail in the next slide about that, but I'm sure you guys kind of remember anyway from way back in the day. And I think they even covered this like early on in class as well. I just really can't remember. But anyway, so it wraps around these histones, and then once it's wrapped around that, these histones and all that can group together like this on this third picture here into what we call a nucleosome. So that's what that is. And then eventually this nucleosome could kind of bound up on itself and form a chromosome. Um, like that's how this happens all the way down here. So I would just know the different levels just to be sure. I would really doubt he would ask anything about solenoids because even I haven't really heard that much about it with my kind of like biochem related background. So I'd be surprised if he ever asks anything about that. But yeah, so I would just know it just in case. But yeah, histones. Honestly, so I would really doubt that he'd have a specific question on histones, but I figured I would include that because I included this last slide just to make sure that you know what histones are, because it is important to know what they are, and that they're kind of involved with packaging. Um, Maybe also just know the names of the histones, but I would say, yet again, probably pretty low yield stuff. Now this stuff, they do like to ask much about histones, so that's another reason why I included the slide about histones here, is the modification stuff that's kind of important. Um, Especially like when they add like an acetyl group, right? So acetyl groups, what it'll end up doing is like weakening the bonds. So this part right here is kind of the more important thing here. So it ends up weakening the bond between histones and DNA when you add a acetyl group to these lysine residues. And that's done by these histone acetyl transferases, right? So that makes sense with its name. It's going to be basically transferring acetyl groups to a specific spot, specifically the lysine residues in the amino acid terminal tail. That's like more specifics. I don't think maybe he'll ask about he'll say something about lysine residues with acetyl groups, but as far as like what tail it's on, I'd be surprised. Um, well, anyway, the whole point is so when you add that acetyl group there, it actually weakens the bond between histones and DNA, and so that makes that whole region of DNA much more open to these regulatory proteins, and so therefore you can have higher level of gene expression in that region. So this acetylation, by picture anyway, so you'd start off with this like big bundle here, but if you add acetyl groups to it and make it all kind of stretch out like this and make the DNA in between the histone complexes easier to get to, therefore higher level of gene expression because of that. Deacetylases, of course, they're going to do the exact opposite. So they're going to remove the acetyl groups and then basically undo this process. And the reason that storage reasons is like why they must be bundled up like that. Because otherwise, over time that would just take up way too much room. Um, if every single histone group like that was separated like that, then you wouldn't be able to package chromosomes as efficiently. So that's why this is kind of like a as needed basis. So that's why you'd add that acetyl group to get to that expression, to get that increased expression via spreading them out. But yeah, so that's it for that. I thought this is also important. Maybe they ask something about heterochromatin and euchromatin. Once again, don't really remember if they mentioned it before because I've heard these terms quite a bit. So it's kind of hard for me to really like narrow down if they specifically mentioned it in this class a long time ago or anything. But the main gist of this is the heterochromatin is like the one that typically is not going to be expressed genes. So it's going to be chromatin that's like in these regions that are just typically not going to be high levels of expression. So you're going to have like the centromere, right? That kind of makes sense because that would be hard to get to, right? Because that would be kind of in the middle of that tetrad structure. Also on the ends, I guess for some reason they have decreased expression, probably just because most of it's kind of like repeated areas down there anyway. Um, but anyway, so heterochromatin, what it says right here sums it up for you pretty much. Devoid of expressed genes, while euchromatin is going to be higher gene expression. So they're like the opposites of each other. So while heterochromatin is not really going to be expressed as often, euchromatin is by far the more expressed. I'd probably also know that 90% of the interface chromosome is going to be in euchromatin. So throughout its lifespan, there's going to be a lot more euchromatin versus heterochromatin. Just because, I mean, it makes sense, right? Because you need all these different proteins in order for uh, the cell's life to be sustained, basically. So maybe keep that in mind as well. Now, I know we did talk about this a long time ago. I decided to keep it in there just in case they were to ask about those subunits or kind of what the purpose of condensing is. But as you can see, right, condensing basically used to condense chromosomes, right? Makes sense with its name. Consists of those two subunits that we've talked about before, so SMC2 and SMC4. And also, as you guys probably remember, or kind of it comes to mind anyway, that the cyclin CDK complex is phosphorylate, condensing subunits early in mitosis, resulting in their assembly on chromosomes. So that whole cyclin CDK complexes kind of activate these condensins to kind of work toward condensing these chromosomes, so that way it can continue throughout mitosis/meiosis in this case. All right, then phases of mitosis. Okay, so we've pretty much, like I said, a lot of the stuff we've kind of covered before, but he does go into more detail about prophase. So this is just, I thought it was important to just know, just a reminder, of all the different phases, so that's why I put a star there for you. 3 essential elements of meiosis. Okay, so you have pairing, recombination, and segregation. So that's 3 elements. Didn't really remember anything specifically coming from this slide, so I didn't put anything, but I thought it was important to kind of understand that there is alignment and synapses and stuff going on, kind of what they mean. So anyway, with pairing, you have one part is alignment of homologous chromosomes with one another. So if you guys kind of remember with metaphase, especially in meiosis 1, you're going to have way more chromosomes, right? You're going to have 46 instead, right? So you're going to have 2 chromosomes per copy or whatever. So you're going to have one chromosome, and then you're going to have an exact other chromosome that's homologous to that. So they're basically the same thing. It's just one comes from the father, one comes from the mother, right? But anyway, what I'm saying here is that those must align with one another, and that's kind of part of that pairing process. Synapsis is kind of when they become closer together, and they're able to kind of exchange DNA. But there is a term specifically for that, so we'll get to it here in a minute anyway. Recombination, so that's actually kind of what I mean with this whole synapsis. So synapsis is kind of that process of coming together, but that recombination is kind of where it exchanges DNA itself. Segregation, of course, that's more aligned with anaphase, or so where they segregate based. I don't know. We'll get to it anyway. It's kind of hard to explain without the slides there in front of you, because the pictures help. All right. So stages of prophase of meiosis 1. So we're in meiosis 1, right? So the first part of it, and then prophase. So you remember how I said there's 5 or 6 different phases of prophase. All of them are testable. So I would say know what the leptotene stage is. So remember that this is the first stage. So remember them in order. That would kind of help you anyway. So I'd probably just write it down in a notebook or something, write it down on the board or whatever you need to do, right? So I would do that, and then put leptotene first, right? Anyway, first stage of prophase 1. What happens in that state? In this stage, right? So you have replication of replicated chromosomes. They condense. So you remember how we talked about condensing? So that's how that whole process occurs. Then you have pairing of homologous chromosomes, and that also kind of makes sense if you think about it with that being early, because it doesn't really take much effort, and since they're already kind of condensed and kind of near each other anyway, it kind of makes sense for that pairing to kind of occur first. All right, then on to zygotine. So that is the second stage. Okay, so right after leptotene. Then you get to, so what's happening here? Synapses are produced through assembly of synaptonemal complexes. Synaptonemal complexes, my bad. So it talks a little bit about this synaptonemal complex. So I decided to include that in there as well because I didn't really remember exactly if they mentioned much about it. But then again, maybe they did and I just don't remember. But what is it exactly? So it binds the homologous chromosomes together. So that's what the synaptonemal complex does. So you remember how in leptotene, right, that they kind of pair together, but they're not actually truly like put together, like stacked onto one another, kind of put together until this whole synaptonemal complex occurs in zygotene. So that's what's happening there. Now it does kind of say here that it also kind of forms during pachytene, which is like your next one. So it's kind of, I wouldn't, see the thing is here, you notice that he says synapses are produced through assembly of synaptonemal complexes. I wouldn't think he would ask specifically which phase that would be in because he said that on this last slide. But if he did, I would probably say pachytene just to be sure. So that's probably what I put in red back in the day. These are actually my slides when I use them. But I know they're the same ones because I download them just to make sure. But yeah, so I'd probably keep that in mind. If he were to ask a specific phase where this occurs, it'd be pachytene instead. But yeah, it talks about what it's consisted of, but the whole purpose is just to kind of put them together. Right, stages of prophase, so continuation. So now we're on to pachytene. So this is where actually homologous recombination / crossing over occurs. Oh, hold on. The slide I got said forms during zygotene. Oh, yeah, maybe they did actually. That's possible. Well, if it's corrected on yours, that's probably the correct one. If they did actually correct the thing, I mean, like I said, I didn't. So whenever I download them to kind of like compare, I don't like compare every single slide, but I'm like, okay, what's the same amount of slides? And they end with the same one and begin with the same one. So I'm like, okay, it's probably the same stuff. And for the most part, it's worked out pretty well. So yeah, I mean, they probably changed it. I just didn't pay attention to it. Yeah. Yeah, exactly. Yeah, you can let me know coming through email or something if they did change it. That'd be nice. Because then I could change it on mine and then re-upload it back to the Google Drive or something. But yeah, that's good. So it's good that they fixed that because I was kind of wondering that when I was taking the class. I'm like, so wait a minute. But anyway, so pachytene. Now this one's a more important stage because of this whole homologous recombination crossing over, which is a very important event in terms of like increasing diversity goes. Um, so that's why I decided to put 2 stars here because you do need to know that that process occurs during pachytene, which is the third stage of prophase. Um, then we have homologous recombination. So we basically just talked about that, right? That occurs during pachytene, but now we'll talk a little bit more into detail on what's going on here. And I've kind of already covered it a little bit back a few slides ago. But anyway, it's used to describe swapping of genetic material between homologous chromosomes within a tetrad. So a tetrad is basically just um, two two chromosomes together, right? If you think about it, that's 4 sister chromatids, so therefore that's your tetrad. So that's why they refer to them as tetrads. Especially now because they've had that whole synaptonemal complex and all that occur at this point. So that's why they kind of refer to this as one single name, a tetrad. But anyway, what's happening during here, as you can kind of see with this picture here on the right, is swapping genetic material between the homologous chromosomes. So very, very important for genetic diversity, right? Because you're switching up genetic material on each of those chromosomes. Therefore, it can kind of increase the number of different possibilities that could arise from, all the combinations of chromosomes with all the combinations of gene swapping. So it's really, really effective in terms of increasing genetic diversity. Crossing over, It's kind of the same thing, honestly. So that's why they kind of put a slash there. So a pretty similar process. Um, I would probably note what chiasmata is on this slide. So the point of physical contact and location of crossing over or homologous recombination. So yeah, they're the exact same thing. But where they cross over is called the chiasmata. So I'd probably remember that. There was like a whole entire slide dedicated to like how it increases genetic diversity, but it all sounded like FYI to me, so I just left it out. But yeah, I would know chiasmata. I would know what stage it takes place, um, and that's in between tetrads too, because I know sometimes those names could get confusing when you're in the testing environment anyway. Diplotene. So you remember those synaptonemal complexes that we formed from, I guess, zygotine. Not really sure. It depends on what chymine says, right, or what your guys' slide says. But anyway, diplotene, the synaptonemal complex disassembles. So happens then. Now it's disassembled after crossing over, right? Because you no longer need them being close together because you've already done crossing over, or homologous recombination, whichever you want to refer to it as. But anyway, now you get diakinesis. So that's where you get like the last phase of, well, actually the first phase of pro-metaphase. So that's the only phase of pro-metaphase. But so diplotene is the very last stage of prophase 1 of meiosis 1, right? And then, so what's happening during diakinesis? I thought it was important to know, so I put in red here. Kinetochores of each homolog become connected to a spindle pole via spindle microtubules. So if you remember right from the rest of um, mitosis or meiosis, they're essentially kind of the same um, on the basic level, right? But this whole diakinesis process, it's kind of like the setup for metaphase, essentially, if you think about it, right? Because they have to all be aligned at the metaphase plate. So it makes sense for these kinetochores to be attaching, for them to attach to these spindle poles, because that's like the next part of the process. But it doesn't really go much into detail for that, because you've already kind of covered it, right? But I decided to include those slides just in case you needed a refresher. But yeah, I'm not going to go over that because we've already touched on it before. Same thing with S phase, so you kind of know what's going on, and then all of this is happening um, in M phase, so you know mitosis, meiosis, all that jazz happening there. But in terms of like where cohesion's actually being done, so if you notice right before S phase, each chromosome is composed of one chromatid, right? So this is just talking about where you get sister chromatids at. So where does that happen? Um, so in S phase, that's where you get from one chromatid to two sister chromatids. But anyway, eventually, when it gets into the M phase here, they get tied together by cohesin, form ring complexes around there, and basically this talks about cohesin. So, once again, doubt they would ask it because it was on that one exam, but I decided to include it once again. Talks more about how cohesin's, how you get rid of it, basically. Yeah, it just talks more and more details about that. Once again, probably low yield, but I decided to include it just in case. If you guys do have any questions about these slides, by the way, that I'm kind of going over real quick, let me know. Once again, I always include my number here at the end of each session, so if you do have any questions about that, you can always text me or message me on Facebook. It honestly doesn't matter. Anyway, so on to the next slide here. So independent assortment. So this is another strategy that's utilized in meiosis in order for genetic diversity to be increased. So what happens here, right? If you remember, in meiosis 1, you have all these different chromosomes. You have the maternal and the paternal, right, chromosomes. So maternal, of course, comes from your mother. The paternal will be coming from the father, right? But anyway, they're not always oriented on the same side. Yeah, I'll hide that. Anyway, when these, let's see, I don't think it's mentioning anything that I need to mention besides what I'm going to say. But anyway, so if you notice from the picture actually kind of helps me out with it anyway. So throughout meiosis, right, you get that breaking apart of all that jazz here, and then eventually you'll have these sister chromatids that like split apart, right? So that's like at the end of meiosis 2. So they'll end up splitting apart to separate sister chromatids. Okay, and so you'll get bits and pieces of each of these spread around. But you notice, right, that you don't have like this green part with this yellow part here, so they're all separated. You don't have any brown with the black because you don't want the exact same copy. You don't want like the same genes just from a different parent in the same cell. Like there's no point to do that. So what independent assortment really is that it's just kind of luck of the draw as to which sister chromatid gets incorporated. That's the way I like to think about it. Um, but anyway, it goes a little bit more into detail. Once again, getting confused about it, just let me know. But yeah, it probably does kind of talk more about that. Yeah, so actually, I think this little quote here probably sums it up for you. So when these homologous chromosomes are segregated, each star cell has a different combination of maternal and paternal chromosomes. So basically what I was saying, but it's just a nicer, fancier way of saying it. Spermatogenesis. Okay. So what I thought was important here, because I asked somebody if it was anything on this slide at all, and they're like, well, maybe. But I would say what's probably important on here is what phase these things are associated with. So primary spermatocytes, right? They undergo meiosis 1, meiosis 2. So that's done by secondary spermatocytes. 2 forms mature spermatozoa. So I would probably just know what phase they're associated with in the order, of course. Picture on the right kind of helps you with understanding like how many chromosomes are within those. So you know, it starts out with 46, goes to 23, right? So that helps kind of get the numbers game stuck in your mind. But I would just know what stage, like I said, with primary spermatocytes, meiosis 1, secondary spermatocytes, meiosis 2, and what's formed after that stage, right? Yeah, I hope it's not unstable. Anyway, oogenesis, right? So basically the same sort of thing what you'd have to know with the last one. So instead, of course, this is the female side of things. So once again, know what's in what part. Also, so the difference here between like males and females is like certain times are like arrested at certain phases. So if you notice, right, the primary oocytes, they're arrested in prophase. And primary oocytes, they're all formed, so there is no oogonia at all once the baby's born, right? There is no more oogonia. It's just primary oocytes from there. And then, throughout that, they're stuck at prophase 1. But however, when they hit puberty, right, you can go from primary oocyte to make a secondary oocyte and a polar body by finishing off meiosis 1, right? And then, if you notice, right, the secondary oocyte, once again, is arrested in a different phase. So it's arrested in metaphase 2 until fertilization, which would complete meiosis 2 and release that second polar body that's associated with the secondary oocyte. So I even have it in the notes down here for you just for simplicity's sake. Know which phases they're stuck in and kind of where they're taking place. That's probably what's the most important out of this. Alright, so now on to the rest of this. So cytogenetics and chromosomal basis. Decided to include this just so you know, because I thought that maybe like that they arrested at metaphase might be important to know. But anyway, what a karyotype actually kind of is it allows you to see like obvious chromosomal lesions. But the reason, so what a karyotype actually is, it's just like a grouping of the pairs of chromosomes, right? So you have like pair 1 here, so the different chromatids is what I mean actually. So you have 2 sister chromatids of 1, 2, 3, so on and so forth, right? So that's what a karyotype is. But also referred to as a karyogram, so they're the same thing. But I thought maybe it was important to know that they're arrested at metaphase, but honestly I didn't really remember too much about it, so I didn't put a star there. Alright, chromosome staining. Once again, not really 100% sure what they're kind of asking or wanting from this, but I underlined the stuff that I thought was kind of like more easier to help you kind of remember the things a little better. Also, once again, didn't know if it mattered if it was where they were fixed at or what was fixed. So they do this using T lymphocytes, and they keep them at metaphase as well. And the reason they keep them at metaphase is just because they happen to be like the most straight kind of drawn out. I think that's probably the reason they do that. Anyway, so they had like G banding, Q banding, and R banding. Tells you about just how, like what the reasons behind them are and how you can see them, things like that. Once again, don't really think it was high yield. May have been like one question on like the different banding, but honestly, I don't really remember, so I didn't put a star there for you. All right, this I do remember them asking a question about at some point about like something being having a metacentric, submetacentric, or an acrocentric centromere. So I decided to put 2 stars on there because I do remember them asking either maybe it was TBL or something, but I really don't think so because from what I remember about the TBL, it's just a bunch of Punnett squares. Okay, so it's probably from the exam. I do remember a question or two coming from this. Probably just one, not two. I wish. Anyway, metacentric. So what is that? The arms roughly the same length. So what that basically means, right? It's right in the middle. Okay. So your P and your QR, they're going to be the exact same. The only time you actually would, I guess, use P and Q is if you had like a shorter and a longer version. So notice here with submetacentric that the centromere is off center, so it's not actually on center at all, and so therefore you'd have P and Q. So P is always your short arm, and Q is always your longer arm. And then you have acrocentric, so it's really, really close to the end of the chromosome. Now, however, this would still be P, and this would still be Q. It would just be, of course, that P is a lot shorter than it normally would be. So acrocentric is really close to the end, but not on the exact end, right? Because you still have to have enough to have that short arm. All right. What I thought was important about FISH is basically what it's used for. So it's used to examine chromosome rearrangements and copy number. It's also typically used for like chromosomal mutation as well. So it's kind of, so if you notice, right, they have like these green and reds and stuff like that. They'll tag, so they have these different probes, so DNA probes, right, in order to like tag it with a fluorescence. So that's how they kind of identify if you have a deletion or not, because of course, if the gene's there, it'll light up whatever color corresponds with the probe, right? So if for some reason whatever that green one is, right, if it got deleted, you wouldn't see a green band. You would just see only the red band here in this case, right? So they're really useful for that sort of thing, and chromosomal rearrangements, right? Because if that green just so happened to move somewhere else and only have that red as well, then that kind of makes more sense of how it could be a rearrangement. So I would just kind of know why it's used and when it's used. But yeah. Okay, now on to the concept of ploidy. So, of course, you guys definitely, I think, would know haploid and diploid, right? So haploid is 23 chromosomes, while diploid would be 46. Polyploid is just more than normal. So that's kind of the way I always think about it. Triploidy individuals, they can be born alive, but they die really soon after. But tetraploidy are immediately not able to have. So they never even get to the zygote stage. So, well, they do, but it just doesn't work very well at all. So they do survive, but not very long after birth. So these can be born alive, but tetraploidy are much worse off. But yeah, so I thought it might be important to just kind of know maybe what the difference in fate between triploidy and tetraploidy were, but from what I remember, I don't think they asked anything about it. Once again, I would include a little more detail about triploidy and kind of how it happens. I thought that was more important, so that's why I decided to include this slide specifically. So dispermy, so that'd be, of course, like more than one sperm gets to the egg. Okay, so I would remember that that's how triploidy happens. So keep that in mind. Non-disjunction of chromosomes. So what happens here, and you can kind of see from the picture here. So non-disjunction. So normally what you're going to be having here is like you'll be having this right here happen, right? So they'll split like this. So you'll typically have, one half goes here, and the other half goes here of this pink one, right? So that's what normally happens. But with non-disjunction, you only have one gamete that gets that, like gets the entire structure. So instead of splitting the two off, right, you only have this one gamete that has all the genetic info. And so what ends up happening, right, is when you go through fertilization here, you get that other sister chromatid, right, from, I guess in this case it'd be maternal, I guess, with at least this example anyway. So you have that maternal here, that blue one, gets integrated into here, and that's how you can get a trisomy from here. Um, so what you'll typically think of with this one anyway, all the time, at least when I think about it, is all the different trisomy, the different trisomy diseases. So you have, of course, trisomy 21, which you guys all know about. So Down syndrome. But that's kind of how this takes place. It's typically by non-disjunction. So all that genetic material only goes into this gamete. Then that fertilization happens with the other half here. And so, but it actually splits off like it's supposed to. But since you have all of that pink already in there, you get a trisomy, and that's how it happens. Same thing over here is going on. But yeah. Also, take note on here as well is where they occur. Okay, so non-disjunction most commonly occurs in meiosis 1 and less commonly occurs in meiosis 2. So I would also keep that in mind as well. So I'll put it in red for you guys. So trisomy 21, as MFM typically goes, its usual fashion. If you see a disease, know it, basically. So know what kind of characteristics it has. That'll help you. If it has a gene and it lists it, that's pretty much golden. That's like the best you can ask for. That's a good day. But in the case with these trisomies, of course, you'd have to remember what trisomy 21 is and kind of what characteristics come along with it. They may also refer to it as its normal name as well, so Down syndrome instead of trisomy 21. But yeah, just in case, I would just know both associated with the trisomy 21 and Down syndrome, along with the other ones as well that we'll get to. But yeah, so know the different characteristics here. I would just pick the things that differentiate them from one another, or the more like obvious signs. So like for instance, we'll get to one that has like a more that I feel like is more obvious and kind of like points out one versus another symptom, right? But anyway, or characteristics in this case. But anyway, know the characteristics for trisomy 21. Know that's Down syndrome. Um, Very important, so I put 2 stars for you there. It could very easily be tested. But yeah. All right, trisomy 18, so also known as Edwards syndrome. The thing I always typically remember with this, and they do use this a lot, is the low set ears and the micrognathia. So that's like a small jaw, low set ears. Yeah, I mean that's typically the things that you see with those people. Also, the rocker bottom feet. Also, it's very like that screams trisomy 18 if you hear any of those. Most of the time, they'll probably tell you micrognathia and rocker bottom feet, or it's like 2 or the 3 of those. They'll probably be on there if they give you trisomy 18 as an option, and you see those 2 or 3 things on there. That's what it is. But that's typically the stuff that's easier for me to remember because it's like super distinguishing from the other trisomy. Oh yeah, yeah. Rich mentioned rocker bottom feet a few times. Yeah, like I said, it's just nice to um, think about stuff that's like super easy to differentiate these. So that's why I typically think of those 3 because mental retardation, right? You already have that with trisomy 21, so it doesn't really help you to remember mental retardation. You also have congenital heart defects as well. A whole lot of these things they kind of overlap. So that's why I typically think of macronathia, low set ears, and rocker bottom feet with trisomy 18. But yeah, they don't have very good prognosis, if you notice from here. So that's pretty much it to know with that. So just know, like I said, the stuff that kind of makes you know it's that. Of course, if you can fit all the rest of that in your head as well, it could help you as well. Um, but yeah, so rocker bottom feet, micronathia, those ears. Okay, trisomy 13, also known as Patel syndrome. So once again, what stuff kind of stands out to me with this is polydactyl. Oh, okay, this one has rocker bottom feet as well. So maybe that one's not the best one, but it does kind of narrow it down for me because I know either trisomy 13 or trisomy 18 has that. But if you notice, right, mental retardation, that's with all 3 of them. So that one definitely doesn't narrow it down for you. But anyway, so microphthalmia, so that's like really, really small eyes. abnormally, I guess, as well. But it's a very small eyes, polydactyly, so um, basically more fingers than would normally be there. And also this cleft lip and palate. Those are kind of the things that distinguish it from the other ones. So I would keep those things in mind. Maybe highlight them things on your slides or whatever. Once again, very low survival rate as well. Probably even a lower survival rate than trisomy 18, but either way, they end up dying shortly after. Anyway, so non-disjunction of sex chromosomes. So now we're going to be talking about non-disjunction with sex chromosomes instead of autosomal chromosomes. So that's what we've been talking about with these trisomy ones. So these are now sex chromosomes. Anyway, all these are high yield, so as all diseases are. Okay, so Turner syndrome. So what is that? That is actually monosomy of X chromosome. So what that means is you basically have an XO phenotype because you're um, but however, these affected individuals are typically female here. So it would be another X if non-disjunction hadn't occurred. But in this case, their actual genotype is X0 or XO. Anyway, characteristics that really stand out to me with Turner syndrome is going to be the short stature, especially the webbed neck and the broad shield-like shape of chest. Those are like the things that really stand out to me about Turner syndrome. Also, gonadal dysgenesis is also a thing with them as well. But the things that really, like I say, stand out for me is that stuff because that's kind of what I see in the picture. So, if you see any of these characteristics, you might need to be thinking Turner syndrome. All right, now on to Klinefelter syndrome. So, so all of these individuals would be male, but they just have an extra X here because, of course, males are just XY typically. But with Klinefelter, you're an XXY, and you have 47 chromosomes instead. So make sure you know this karyotype with it. Um, anyway, the things I like to think about with Klinefelter, you're typically going to be thinking gynecomastia is a very big one. So that's one thing that really stands out to me. And along with that, you can kind of group all these three things together. At least in my mind, I kind of do, right? Gynecomastia, obviously they kind of have like lower testosterone, if they're able to get breast development. So therefore, I kind of tend to think hypogonadism, lower testosterone, and also if they're able to get breast development, then maybe they're just having problems with the secondary sexual characteristics. So I kind of group all those things together. Once again, I kind of go along with gynecomastia here, um, and with the tall and thin stature. Although that doesn't really differentiate you from Marfans very well, it does kind of help me specifically. So if it helps me, it may help you. But so that's what I'm trying to do with all these, just kind of hit the I would say the higher yield or like the easier things that kind of help you remember what syndrome is which. Um, but yeah, pretty much all of these are high yield because you never know, they could switch it up on you. But I thought Turner syndrome may have been on there specifically, but I'm not too sure. This slide, I don't really remember if they mentioned any of these two, but I did put a star here just in case they could come up. So trisomy of X, so of course that's going to be triple X here. Usually it does not show an abnormal phenotype. Yeah, it kind of tells like more about that. But I would say because this stuff's kind of, I guess the way I would kind of remember this is maybe the intellectual delay and the taller than average. I guess you can put those two together because Marfans and Klinefelters, they don't really have mental differences on them, so maybe that could help you. And also, um, they're female, so it couldn't be Klinefelter either. Then you have males, so XYY syndrome. Going to be taller than average, so once again, it's another taller kind of thing. I would probably, with that, I'd probably learn the ADD hyperactivity type thing because that's kind of distinguishes them from most other things. So maybe that might help you. But once again, I don't really think they asked any of these, but I put a star here just in case. Chromosomal translocations. Okay, so we're talking about like now specific, um, I guess, changes within chromosome structure. So he does go a little bit into that, so I decided to include most of these. But anyway, so what the difference here between translocation, if you notice from the picture, right, is like bits and pieces of chromosomes being like exchanged with another chromosome. So if you notice, right, chromosome A and B here, they switch these parts with one another, and you have that translocation. Right. Now, the thing to notice here is you have balanced and unbalanced translocations. So with balanced translocations, you don't have a gain or a loss of chromosome material. So that's more like this top example. So they're, variably like, um, I guess like the same size or something. Like you don't lose any material because you're exchanging this material and putting it onto the other chromosome, and you're exchanging this material, putting it on the other chromosome. So you're not losing any material. You're just exchanging it, right? Now with unbalanced, if you notice from this example, that take this bit that you had up here with balanced translocation, and you basically put these bits together. So say you take this like purple one here, and you take this mixed hybrid one here on the right, and put them together. That would be an unbalanced translocation because you've lost that bit of pink right here, right? So notice it's no longer there because it's back on this piece here, right? And since you can only have, that pair together, you lose that material. So you no longer have that bottom part of that pink part. It's no longer there. So therefore you're losing material, and therefore it's unbalanced. So definitely not a good thing for sure. Translocations in general are not good, but that would definitely be bad, is if you're losing material. So now he goes in like specifics about like examples of translocations. So the one I think that's always important every time they mention it, BCR-ABL, they mention it all the time. Otherwise known as the Philadelphia chromosome. It's associated with CML. So CML is pretty high yield with BCR-ABL. I would also just know that they refer to that as the Philadelphia chromosome, and that's in between 9 and 22. That'll kind of actually help you later on with heme because it does come back with CML. So if it helps you, you're kind of honestly with all this stuff. I mean, you're already like board studying way ahead of time if you just remember it, right? Anyway, so BCR-ABL, remember that's associated with CML, 9 and 22, and that's it. So Robertsonian translocation. So this is, I guess, a little different than what you typically think of with a translocation because it's just a little odd. Anyway, so in a Robertsonian translocation, the short arms of two non-homologous chromosomes, so this would be chromosomes that aren't typically paired together, right, are lost, and then long arms fused together. Okay, so that would be cutting off this top part here and the bottom part of this one, right, and that entire fragment's gone, but then you only have the long arms of both of them combined into one chromosome. And of course, that has to happen between two acrocentric chromosomes because that's the only way you could have small enough fragments in order for them to be lost. Because if it was like a submetacentric chromosome, then there's still like a relatively long P arm. So you wouldn't be able to just basically lose that fragment. But anyway, people that have Robertsonian translocation, they of course, since they're losing essentially one chromosome, right? They only have 45 chromosomes. So that's something to keep in mind. Robertsonian translocation. I put 2 stars here because I think they may have had a question regarding Robertsonian translocation. So I'd probably keep this basically what is a Robertsonian translocation, that they have 45 chromosomes, and what kind of chromosomes this typically occurs with, so acrocentric chromosomes. Also, a thing I thought that was important is that Down syndrome can also occur due to a Robertsonian translocation. I don't think you really have to know like the specifics like in this bottom part here, but I would just know that Down syndrome can also happen due to a Robertsonian translocation. All right, now chromosome deletions. This is a much simpler thing to talk about, right? Because you're just basically deleting a part of a chromosome. So put it in here just to have like all the different segments for this. All right. Now on to, so once he gives this kind of slide, He gives like examples. So crew to Kai. I think this one might have actually been on our exam when we had it because I think I like it's just one of those memorable ones. So I feel like it may have been on there. Um, but anyway, once again, with all these diseases, know that it's a deletion. So that's something I kind of didn't really stress too much. So when they do give examples, they may ask you on the exam like what kind of mechanism caused like this disease or whatever, right? So they could give you all the like the symptoms or characteristics of crew to Kai, right? But then they don't actually ask you which disease it is or disorder. They ask you instead what mechanism led to this disorder. So that's just an example of a question they could ask you. So that's why I decided to include that slide along with it, so you know what's going on and you can kind of organize it better in your head. Okay. Anyway, the things that make this one stand out is definitely the laryngeal defects. Um, so that's why it's called that. So cry the cat is because it has like a cat-like high-pitched cries. Now on the exam, they're probably not going to actually mention cat-like because that would be like obvious. They wouldn't want to give that away to you, but they definitely could mention something about like their voice being high-pitched or something like that. So keep an eye out for that. Also, microcephaly kind of stands out to me specifically, um, and also intellectual disability. They only list pretty much only three things on this slide anyway. But yeah, laryngeal defects is like the number one thing. Oh, also thing of note as well, I would probably at least keep in mind like what chromosomes kind of involved. I mean, I think especially like with this one that we'll get to, the George, that one they like to ask you about because I mean they even mentioned 22q11 deletion syndrome. Um, but yeah, so maybe it wouldn't be a bad idea to remember the chromosome, but do I remember them really asking much about that? No. So it's kind of like on the lower yield of things, but if you have the time, by all means. Wolf Hirschhorn, or I honestly don't know how you say that one. This one's also high yield as well. Deletion occurs at 4p16, very close to the telomere. The characteristics that kind of stick out to me is I would say maybe the skeletal and dental abnormalities here. So that kind of stands out for me. Also, you notice that they mention like specific facial features here. So those also kind of help you differentiate sometimes. So maybe like the widely spaced eyes or the broad flat nasal bridge, because they will mention like a facial characteristic as well. Typically, if they give you this as like an exam question. Also, another thing, maybe weak verbal skills. Like try to just think, like I said, the things that differentiate them from something else. All right. 22q11 deletion syndrome. Otherwise, like I mentioned earlier, the George syndrome. Um, Things I think would be important would be this. So, defective embryonic migration of neural crest cells to developing structures of the neck. That's probably pretty important to know. So, I have that highlighted in red for you. Also, I would remember that there's functional deficits of the thymus, and that since you have functional deficits of the thymus, you would have hypoparathyroidism and hypocalcemia. I would keep those in mind. Um, More than likely, if they're going to give you the George, they're going to ask you about this sort of thing with the whole thymus and cleft palate, that sort of thing. As like these three are like the more important ones for sure. Also, since so many of them have congenital heart defects, maybe they mentioned something about that as well. But yeah, definitely the thymus thing is a big important thing to do with this syndrome. Yeah. Now, Williams syndrome, okay, so it's a microdeletion syndrome, like all these other ones, they're deletions, okay. Now, what makes it stand out to me with these? So notice that they mention a specific like genes here. I would probably know these different genes. So I probably know ELN and LIMK1, so LIMK1. I would know that, would have something to do with Williams syndrome. So that way, just in case they mention it, right, that those have something to do with Williams syndrome. So if they have this in the question stem, ELN gene or LIMK1 gene or protein, that's kind of like a dead giveaway for Williams syndrome. Okay, but things that kind of like stick out for me is the poor visual spatial abilities. So that's something, and they even mentioned it twice if you notice, right? Visual spatial deficits, poor visual spatial abilities. So maybe keep that in mind. Also, the large mouth kind of thing. You don't really see that often with all the other ones, so that might help you differentiate it. Right. Chromosome duplication. So instead of a deletion, now you're actually getting like an exact duplication of that genetic material. So pretty simple from this explanation of this illustration here. But yeah. So now on to duplications. Now Charcot-Marie tooth. This is like super like something they probably will ask you about because I think they may have asked it on our exam. So that's why I put 3 stars with this one. But anyway, what gene is associated with it would be PMP22. Definitely know that. Probably not important to know the chromosome. Definitely the gene. So no PMP22. Also know that this is like a demyelinating peripheral polyneuropathy. So just in case they mention any of these symptoms here. So if they have like slow progressive muscle weakness, atrophy, upper extremity ataxia/tremor, then you need to be, and if they ever mention PMP22 gene, that's Charcot-Marie too. Okay. But yeah, reason I put 3 stars because I'm pretty sure they've had this one on our exam. Chromosome inversion. So there's like a difference between paracentric and pericentric. Anyway, so paracentric, it does not include the region of the chromosome within the centromere. So notice like it doesn't include this middle part where the P arm and the Q arm meet, but instead some other region, right? So it just basically flips it. But with a pericentric, it includes the centromere. I would say that's probably important to know. There may have been like a question with that, so maybe that's where something like that came from. But yeah, so notice there's no loss of genetic material at all. That it's all balanced out. They just flip it over. All right, hemophilia A. Okay, so that's an example of a chromosome inversion. Basically, what I would know there is that you have mutations in the gene for clotting factor 8, and that's X-linked recessive. You have to definitely know it's X-linked recessive anyway because of the whole genetics chapter. So that lecture, you have to know it for that anyway. So just probably go ahead and stick that in your head. Anyway, things to know with that. You have like severe bleeding from wounds, hemorrhages in joints and muscles. Basically bleeding everywhere is the way I like to think about that. Ring chromosome. I think they may have had a question to deal with ring chromosome. So I decided to include 2 stars with this one. So what symptoms you find, right? You have seizures, intellectual delay, all of these things I've read for you there. But yeah. So what happens there is you actually get deleted material and you form a ring chromosome. XP isochromosome. I only include this slide because some of Turner syndrome can happen because of an XP chromosome. So I decided to keep that, just like how I kept Robertsonian translocation, right, for Down syndrome. So same reason why I kept this one. All right, so now on to the next one. So autosomal dominant and recessive inheritance. Okay, so basically I kept this slide just in case you don't know like the basics of a pedigree. But anyway, I'll just kind of break it down real quick. So males, they're going to be always squares on here, while females are always going to be circles. All right, and these lines on each of these pairs here. So you notice in between 1 and 2, that would be kind of like their mating, right? And then down below that line underneath them, it's going to be their offspring, and so on and so forth. So that's what's going on with these. Now it does kind of break down other bits that you have to know as well. So it does help you with this little chart here. So notice that the slash through it means they're dead, so they're no longer alive. Right? I doubt the twins thing would matter. I doubt that would even come up with here. But yeah, it's important to know how to read these. So I decided to include that. Also, I decided to include this as well because it kind of gives you a nice little basis for dominant recessive alleles. But basically, dominant is pretty much the phenotype. So the dominant allele is the allele that expresses the phenotype regardless of whether there's two copies or one. So basically, the example I like is this down here. So in this case, this big B, right, that's your dominant allele, while this little b, that's going to be your recessive allele. So, and this is actually rings true um, in real life anyway. So brown eyes is definitely dominant toward the recessive blue eyes here. But anyway, so if that big B is there, regardless of if there's a little b or not, it's going to be brown regardless. Right? But the only way that you're going to have blue is if you have both of them are little b, little b. Right? That's the only way you get it. So that's why it's recessive, and the other was dominant, because if it's ever expressed at all, it's going to be that phenotype. But if it's not expressed at all, then it's going to be recessive. All right. Now, once again, kept this one because I thought it was like a nice basic illustration of what a Punnett square is. I did kind of mention earlier how this exam was like all about Punnett squares, like not the exam, but the TBL was like all about Punnett squares. So be sure to kind of like understand the basics for sure. Um, I think they go more into detail on like the inheritance patterns later. They do kind of with this one, um, but yeah. So just kind of know how to do Punnett squares. If you have any questions at all about Punnett squares, let me know. I can kind of walk you through some of them if I need to. But yeah, basically what you do, right? So say you have a parent here that has BB and a parent here that has like little b, right? You cross them together here, so you put one parent this way, one parent this way, and you do like a little chart. That's pretty much how you do it, right? And what they're going to typically ask you from Punnett squares is like what percentage of offspring from this pairing will have whatever disease. But in this case, right, if you were to ask a question about this, they would ask you what percentage of the offspring will have blue eyes. Right? Let's say, for instance, that's the question. Then it's 50%, or 2 out of 4, or 1 out of 2. So they could ask you a fraction instead of a decimal. But either way, that's kind of the way they'll ask you questions with that. But that's like a super simplified example. Um, anyway, so then I decided to include these slides as well because they kind of help you understand the different inheritance patterns. So with autosomal dominance, it doesn't matter if your offspring is female or male, it doesn't matter at all, and there's no skips in generations whatsoever. So pretty much every single generation is going to be affected. And so you can actually see that with this pedigree, right? Because this, of course, number one, that lineage has one, then two, that lineage does, then three, so on and so forth. Every single lineage or generation would have somebody gaining that disease or disorder, right? And it can include father to son, but not specifically father to son. But what's really helped you is, so a lot of these differ on if males are affected or females are affected. But of course, autosomal dominant, right? It's going to be both are affected, so it doesn't really matter at all. Okay, so autosomal dominant includes every generation, and it's not picky. Okay, so it's going to affect everybody. Now it gives you examples of autosomal dominant diseases. It is important to know what diseases are autosomal dominant, which ones aren't, because I'm pretty sure they ask that sort of thing. So they'll have like Punnett square questions on TBL, but they'll also have like what diseases are what. Also, so the things with TBLs as well that I remember them doing is they could give you like characteristics of achondroplasia, but then you need to remember in your mind, hey, achondroplasia, that was autosomal dominant, right? And so therefore, since you know it's autosomal dominant, then then you can kind of fill out the Punnett square from there. So that's kind of the way you need to be thinking about these, because that's kind of the way they ask about it on TVO. Yeah. So just know achondroplasia is autosomal dominant. Make sure you know all of these diseases and which one they are. So autosomal dominant, yep, achondroplasia. Also, with achondroplasia, know this GLY, so glycine 380 arginine mutation. So know that part. as well as what genes affected with that. So FGFR3. That's very, very important to know for achondroplasia. Also, that this mutation, this GLY38ARG, right? That's a gain of function mutation, and therefore it's like continual activation of a receptor, and therefore that. But what happens, right, when you have that receptor continuously activated, is that you have this inhibition of chondrocyte proliferation. So basically, you won't form correct cartilage, and therefore you won't form correct bone. So it won't be long enough. Therefore, all your long bones are typically affected. So that's why you get this sort of phenotype here. Um, Also, make sure to note that individuals homozygous for that mutation. So the only ones, so they do not live. So if you actually had big A, that's actually lethal. So those people do not live. So make sure you know that as well. I actually kind of like this diagram in the bottom right. Kind of helps you with that. Once again, always know the symptoms associated, but I'm sure most of you guys would probably know what achondroplasia would look like, right? Marfan's. So Marfan's, autosomal dominant disease. What mutation is it? Fibrillin 1, so FBN1 gene. Um, what does that really do? I don't think they really ask much questions about this whole TGF beta thing, so I probably wouldn't. I have that in red just in case they were to ask that, but I don't remember them asking specifics on that. All right, now the things that really distinguish you from our fans is tall stature, of course. That one's always got to be there because that's like the obvious thing with Marfan's that you kind of think of. Also, the aortic root aneurysm or dissection. I think that's the one they decided to include with our exam. But they could also do the ectopic lentis. I don't think they really ever mentioned the arachnodactyly, but I guess they could. But yeah, they're all kind of like, they sound totally different from other diseases. So typically, Marfan's is not one of those you're going to get confused because it's like totally different. And I put 2 stars there because I felt like it was super important to know. But yeah. And then neurofibromatosis, once again, that's another one you need to know. And notice, once again, just as a reminder, these are all autosomal dominant, so make sure you know that for the TBL and for the exam. So NF1, that's going to be your mutation. The things that really super stand out about this, okay, is this cafe light spots, I guess is how they say that. I'm not really sure. But pretty much every time they mention it on the exam or anywhere, they mention these spots. Okay, so that's super high yield in terms of neurofibromatosis. But typically, you just always think of like spots and tumors with neurofibromatosis. That's the way I think about it. And it helps me. Um, yeah, so there is like, so the thing I mentioned about like Marfan's as well as neurofibromatosis, they have an example part here. I would also know that this is an example of pleiotropy, and that this is an example of variable expressivity. Now, it's not really going to make any sense to you right now because I think Zegrom kind of goes more into detail of that whenever she goes, like whenever she has her lectures. She'll talk about these different ones. So pleiotropy and variable expressivity. But yeah, just kind of keep that in the back of your mind. Familial hypercholesteremia. Okay, so this is another autosomal dominant. So what's going on here? You have a mutation LDLR. Okay, that of course leads to high levels of LDL because you're not able to take it up with the receptor anymore. Okay, because we've already talked about this one anyway with the Kicks material from from a couple blocks ago. So I'm sure you guys know all about familial hypercholesteremia, right? Things to note, of course, is the accumulations of lipid under the skin. So, or these xanthomas, which is basically what those are. So that's what xanthomas are. It's accumulations of lipids under the skin. But yeah. So know that that's autosomal dominant. Know the mutation. Know the xanthomas. Retinoblastoma, once again, it's autosomal dominant. So we're still in that category. Remember that's RB1. And also that this is an example of reduced / incomplete penetrance. So that's also one of those things you need to keep in mind. All right. Then on to this part. So I didn't think it was like super important. I put a star here just in case it was. Um, but typically, cancer syndromes, you don't really tend to think of it as being an autosomal dominant inheritance pattern, but it can. So I thought it was kind of important to maybe just like, I guess it's an important FYI. I don't think it's like super important. All right. This slide's always nice. So these slides, when you get to them, I decided to include them because it's like a quick, easy way to like remember. So if you can come up with like nice, easy ways or something to remember all of these and that they're autosomal dominant, it would help you out a lot. Because especially for that TBL, because like I said, they do um, give you like the characteristics of the disease, so you'll know that, but you may not know the inheritance pattern in order to do like the Punnett squares or whatever. So make sure you know that, and that all of these are autosomal. Autosomal recessive. Okay, so now we're on to recessive instead. Since it's still autosomal, all males and females are equal, so everybody gets it. If they have, of course, if only if they have this little a, right? That's the only way you get it. So that's why you don't get it every single generation like you would with autosomal dominant, because it just happens to have to be little a, little a. Okay. Now, cystic fibrosis. That's the first example it gives you. I thought cystic fibrosis was very high yield, so I'll put 2 stars for you there. Also, you know how they keep showing all these examples here? This is an example of allelic heterogeneity. Okay. Um, Logan, I emailed him about those diseases. He said we had to know all of those, even though he didn't mention them in detail on the slides. Just a FYI to those in here. Oh, okay. Yeah, I do kind of remember him. I was thinking that too when I got to that slide. I was like, I think he even expected us to kind of know acute intermittent porphyria somehow. So maybe just kind of keep that in mind. But yeah, they expect you to. Yeah, exactly. Yeah. So I mean, it's nice to stay on the side of caution, just in case. Um, Now, do I really remember him doing that much on the exam though? Not really, because there's like so much that he could ask you. Yeah, but he could do that with TBL. He does like to do that with that. So I would keep that in mind. But yeah, the exam, I don't think he really ever. He was asking stuff that came from this block. He didn't really ask anything from other blocks. This exam, if you haven't already noticed, like I've mentioned before, it's all diseases. So it's one of those things. If you have like a good Anki, which I think I may still have mine. If I do, I'll put it on the drive that has like all the diseases on it. Um, but of course, that would actually be more for the cumulative. If I have that, I'll put it on here. If not, I can probably find somebody that has that. So that's just a heads up for cumulative. Um, Now, in terms of this exam, I actually do have. So there's this form I'm going to put on there. So I actually made it last year for all the diseases of this block. Personally, I wasn't a big fan of Anki back in the day at all. So I made my own list. I was going to make my own list of all of them, but it took way too long. Didn't have enough time. So I do have a list for all of the Block 7 diseases, though. So I can definitely hand you guys that. Put it to good use. It took a while to make that, by the way. But it does look, I think it looks rather nice. Easy to look at. It's easy on the eyes. Alright, so now on to the rest of the stuff. So I was over recessive. Right, cystic fibrosis. Yeah, no problem. No problem at all, Teresa. Okay. So mutation CFTR. Typically, you're going to be thinking of that as like a chloride transporter. Um, I don't know. I thought they had. Did they ever have a TBL regarding um, like, have they given you guys like a article about cystic fibrosis or anything? Has that been a TBO yet, or anything to do with a TBO? Because I felt like that was something one time. Oh, okay. Well, maybe they will with this one. Okay. So if they do, cystic fibrosis was pretty important because that entire um, article or whatever was about it. So just a heads up in case they do that. But I'm pretty sure we had one, but maybe it wasn't for this. Maybe it was for some other class. I don't know. But yeah, just curious. Tay-Sachs disease. So you guys have actually already seen this before. Things that are like more important is what's bolded. What's in red, that's more important on this slide. So hex A or hexosaminidase A, that's your deficient enzyme there. Note, of course, that's autosomal recessive, and that it has this cherry red spot on the retina. Now, the only I usually wouldn't mention that much because there is, so Niemann Pick also has cherry red spot, but since they don't really mention Niemann Pick at all, I would just remember the cherry red spot for this exam. It honestly helps me differentiate it pretty well. But anyway, also thing of note here is that if you have no hex A or a deficiency of XA, then you have an accumulation of ganglioside. Therefore, that leads to neuronal cell death within neurons. Once again, this slide's important to know for TBL for exam purposes. So know it, and it sums it up for you. And even like he was saying earlier, even the ones that we've already had before, I would also keep that in mind. All right, dominant negative effect of P53. Honestly, don't really remember if this was super important or not. And this slide is a little confusing. But anyway, it's kind of explained if you read through it a little more. It explains it pretty well. But for quickness sake, I'm probably just going to skip over it so we can get through all these different diseases. I can kind of point out what's important. But if you do have any questions on this dominant negative effect of P53, just let me know. I'll go through it with you. All right, so beta thalassemia, that can be actually either dominant or recessive. But this is one of those things that I don't think they really ask much about. And the only reason they kind of have this here is because of this whole dominant negative effect. So maybe just know that that's associated with beta thalassemia, but I think it's pretty low yield, but I include it just in case. New mutations. So the thing about new mutations, instead of germline mutations, which are passed down from generation to generation, new or spontaneous mutations, they arise in its own generation. So it hasn't been passed down at all. It's brand new. Okay. Now the thing to notice, right? If a mutation occurs, so they can be transmitted from an unaffected parent to an affected offspring if it occurs in a germline of the parent. Yada yada yada. Basically, just telling you about new mutations. Not really important, but just in case. Factors influencing phenotypic expression in single gene. Honestly, I think I honestly just forgot to delete this one. I don't even think this is all that important. Oh, wait a minute. Oh, this is actually kind of a useful one in a way. So they do kind of mention the pleiotropy with Marfans. So they do kind of tell you the phenotypic expression single gene thing. So you remember how I was saying know the examples of these different things they've mentioned? So maybe pleiotropy with the Marfans and so on and so forth with these different ones. Alright, single gene disorders, so now sex-linked dominant and recessive. So these are all going to be sex-linked instead of autosomal. So this kind of goes into details about X and Y chromosomes, kind of where they come from. Things to note that I thought was kind of important is that the sex of the offspring is determined by the male sperm, just in case you guys don't know that. And that's the reason is for that is that the females can only give an X chromosome, but since a male has an X and a Y, they can give either one, right? Now, and another thing to note as well that kind of helps you make sense of the inheritance pattern for X for sex-linked chromosomes, for sex-linked disorders, is that the X chromosome in every male was inherited from the mother. Okay, so just keep that in mind. It's a pretty big thing to keep in your head. Okay, the X chromosome. So every time that a male has an XY, right? They have an XY. That X comes from the mother. So if the mother has a disease on that X, so if there's something wrong with one of her X's, and she gives that X to that male offspring, he's stuck with it. There's nothing he can do. Okay, but if it's given to a female, it's no big deal because they have another X from the male, right? that doesn't have that disease, unless the male just happened to have that too, I guess. But what's the chances of that, right? It's like 1 in a billion. So, yeah, that's something to keep in mind. So, therefore, any X-linked condition will manifest in male. A lot easier than female. Okay? So, X-linked dominant inheritance. Okay? Seen twice as often in females. Now, why is that? Because it sounds like it would be going against what I just said, right? But in this case, since it's X-linked dominant and you have 2 X's with females, right? Then it's going to be seen a lot more often in females because the chances of getting that X is a little higher because they have 2 X's, right? Another thing to notice as well, all daughters of males that have that disease get affected. And why is that? Because they have that X and they have to give an X from them and an X from the mother as well. So therefore, if that X that they're giving is affected and it's an X-linked dominant inheritance, then they're going to get that disease, right? But notice that there is no male-to-male transmission. Now, why is that? Because in that case, right, the male that would be affected by the disease, he would be giving the Y instead of the X that's affected, and so therefore that male would not have the disease. So it's just it helps you a lot when you think about it in that sort of circumstance. But yeah, so read through this a couple times. Go back to this last slide if it's not making much sense to you. Always though, if you have any questions about it, I'm always free to help. All right, Rett syndrome. Okay, this is an X-linked dominant disease caused by mutations in MECP2 gene. Okay, maybe know what MCEP2 is associated with. That's always something that they kind of like to ask about every now and then. Also, know the symptoms that kind of stand out for you. The things that stand out for me is ringing or flapping movement of hands. I think they may have actually said that on ours. Maybe the seizures and the spastic ataxic, because that kind of stuff kind of mixed together in my mind. So maybe keep those in mind. But of course, any of them could be included. But yeah, so know that this is MECP2 associated, Rett syndrome, X-linked dominant, and some of the symptoms. Vitamin D resistant rickets. The only reason I included this was just because it happened to have a gene associated with it. And I was like, well, if it has a gene associated with it, maybe it's testable. And like what it codes for, what's associated with it. But honestly, do I remember them asking anything about this? No. So I didn't put any stars on it. All right. Now, this whole page sums up all of your X-linked dominant disorders. Not very many of them, right? So you have that vitamin D resistant rickets, you have Rett syndrome that we just talked about, and you have Fragile X. Now, Fragile X, I think, was actually mentioned at some point. Now, it seems very, very, not important, from this slide, but it actually was. So I would definitely keep this in mind. So know the different the symptoms or characteristics of this, because it will help you out. All right, now on to X-linked recessive. So this is what you typically think of as X-linked inheritance, but the other one's a little different, and that's why there's not that many of X-linked dominant versus X-linked recessive. There just happens to be way more of them, so that's why you typically think of sex-linked as an X-linked recessive. But anyway, the thing to know with these is what I mentioned a long time ago with that intro slide, is that since the males are going to be getting that X from a female, right? They're the ones that typically get affected if that female has that, right? Has a copy of that whatsoever. Okay? So that's something to keep in mind. So therefore, affected fathers, okay, will pass on that mutation to all daughters who will be carriers. So this one right here, right, this male, since he has that X that's corrupted, he's got that disease, and he has that normal Y, every daughter that he has will gain his X, right? And so therefore, they won't have the disease because it's X-linked recessive now, but they'll still be a carrier. So that would be their phenotype would be this up here. I mean their genotype, my bad. Also, so the only time that they typically get affected, females, with X-linked recessive is if they have X inactivation. So if, for instance, say you have a carrier, right, and that X that has that disorder on it, say that one's the one that's actually being used and the normal one is inactivated, you could have symptoms. But it's probably not typically as bad as the full-blown disease itself. So that's something to kind of think about. Now, thing affected males inherit disease through females. So I've kind of already covered that point, but yeah. Now on to royal hemophilia A. So a little different. Okay. So royal hemophilia A mutations associated with clotting factor 8. Okay. All affected individuals are male. That's something you probably need to keep in mind. So that's something that just kind of stands out to me. Also, of course, since it's a hemophilia, you're going to be getting like bleeding everywhere. Muscular dystrophies. So this is also a X-linked recessive disease. Now there's two different forms. There's Duchenne muscular dystrophy and Becker muscular dystrophy, so DMD and BMD. Main thing with muscular dystrophy is you have a progressive weakness and loss of muscle. It's X-linked recessive, therefore it predominantly is in males, of course. I would probably know in terms of being in between Duchenne and Becker, so know the differences between those two. So notice that they're both mutations within DMD gene, but Duchenne, it's typically going to be known as like the much worse version of muscular dystrophy. And it kind of even, in the way I think about it, is because it's due to a worse mutation. So it's due to a frame shift mutation, which would basically mess up everything from that mutation onward in that gene, right? Versus Becker is not caused by a frame shift, but instead like a deletion or something like that, right? And so therefore, since this frame shift is happening in Duchenne, you're going to basically have a truncated, non-functional dystrophin protein. So you're not making any dystrophin that works whatsoever. Okay? So therefore, it's automatically degraded, and you have no dystrophin protein, and therefore you have a much more severe phenotype. Okay? Now with Becker, since it wasn't a frame shift, and therefore it's not as much of a severe mutation, you still get dystrophin protein made, but it has reduced function. So it still works, and it's being made, so therefore it's not being degraded, and it has a purpose still. So therefore, its phenotype tends to be less severe than Duchenne. So all of that stuff, remember that. Remember the differences, remember the mutation, remember that's autosomal recessive, so therefore it tends to happen more in males, that kind of thing. I thought this was also kind of an important slide to include as well, just because like it does kind of go a little bit more into detail as to what muscular dystrophy sort of looks like what kind of signs they have. This Gower maneuver is very important. They talked a lot about that in MSK. It goes back and you see it, he's like automatically like, oh, muscular dystrophy. Anyway, that's pretty much it for DMD or just muscular dystrophy in general. Color blindness. So this is X-linked recessive. So therefore it's more common in males, right? What I thought was kind of important is that it's red and green cones are the ones that are kind of affected. So maybe keep that in mind. So therefore you have deficits in red or green perception defects. I think that may have been a question, so that's why I put a star there. Christensen syndrome. So that's actually, if you remember back to Cooper's Carnival, that's the one that is inspired by. So that's Christensen syndrome is the so that's what Cooper's Carnival is kind of awareness for, Christensen syndrome. That's also X-linked recessive. It's caused by mutation in SLC9A6 gene. Okay. What is that code for? Codes for this exchanger or this channel protein. I put 2 stars there because somebody actually told me that they include a question on Christensen syndrome just because of like Cooper's Carnival and all that jazz. So I decided to include 2 stars on that one just in case they would ask something about it. Now, do I remember it being actually on that exam? Not necessarily, but it could have been on there. Just remember all the symptoms, of course. Know the gene. Know that it's X-linked recessive. These are the same usual stuff. Once again, these slides are always gold, okay, because it sums it up for you, helps you realize what's X-linked recessive, what's not, so on and so forth. Now, this I put 3 stars on because this thing helped me out a lot to keep these things straight, okay? This picture's awesome, okay? And you can follow like this little diagram, and it helps you kind of keep in mind whether that person would be affected or not, you know, kind of like what's going on. So I really, really like this diagram. You know, keep everything straight in your head. All right. So X inactivation. I actually talked a little bit about this already. It just goes into more details about it. Do I really remember a whole lot about X inactivation being on the exam? No. But once again, this is kind of one of those things. If you have any questions about it, let me know. Mechanisms contributing to X inactivation. So the thing I will mention about that is this XIST gene is kind of what's like responsible for all of this. So once this XIST gene is activated or expressed, it's going to produce an RNA product that coats the chromosome and helps produce its inactivation. So that the RNA product from that gene being expressed is what coats the chromosome and therefore inactivates it. Okay, so that's why I thought it was more important to know that, and put 2 stars here for it. But yeah, unique characteristics of mitochondria. The only reason I included this slide is just to kind of give you characteristics wise of like what mitochondrial DNA looks like. So it's circular in shape, so it's got like a plasmid shape instead of like regular DNA, of course, which is like, you know, double-stranded helix type thing. And linear. But anyway, the thing to know about mitochondrial DNA is that all of the mitochondria in an organism originates from the mother. So therefore, if the mother has that disease, it'll be passed down to the offspring automatically, right? Because that's the only DNA mitochondrial wise that gets passed on. So therefore, if the mother has this mitochondrial disease, okay, if that DNA is affected, then the offspring, no matter what their sex, gets affected. Okay. Mitochondrial inheritance, like I said, I've already kind of covered this anyway. The only thing that I thought was kind of like something to note here, and I don't know if it even mentions it on this slide. Actually, I already kind of mentioned some of this stuff. Okay, so this, I feel like honestly is probably kind of like FYI information. I just thought it was kind of neat to include. So I've kind of already talked about this. So mitochondrial disorders can only be passed from mother to child, not from father to child, because that's where you get your mitochondrial DNA is from the mother. If a female has pathogenic mitochondrial DNA, and I actually just said this, so that nice little red thing sums it up. All right, heteroplasmy, okay, so it's uneven distribution. So what is that? Uneven distribution of specific mutation in mitochondrial DNA between daughter cells during cell division. So the thing to note that's important is that some cells may inherit more normal mitochondria, while others inherit mostly mutated mitochondria. Now, if you remember right from the structure of a cell, that there's like multiple mitochondria within a cell, right? So it's not like your genome, right, that like is only in the nucleus, so there's only like one copy of that per cell. With mitochondria, right, you have multiple mitochondria, therefore you have multiple like different mitochondria with the same DNA, but could be, you know, different. Like could have different expressivity and everything, right? So therefore, yes, you could have variable expression of mitochondrial diseases because there's more than one mitochondria. But the thing that's important to note is that the larger the percentage of mutant mitochondrial DNA molecules, the more severe the expression. So the more mitochondria that have that mutant DNA, right, the more severe the disease is. Very important concept to know. This kind of gives you like more of a breakdown of how it kind of works. So the thing here is that the mitochondria with the abnormal DNA is red, so therefore the more red it has, the more affected the offspring is. So notice that, of course, if it arises from this germ cell with like this kind of mitochondria in it, then it's going to be very affected. And of course, this will be unaffected because it has less red, and this has like middle amount, so therefore it's going to be like mildly affected. So yeah, mitochondrial diseases. Just talk about like how they have like common symptoms. I thought that was kind of interesting, so maybe note that. It does go like in the specifics about like these different diseases. I just thought it was kind of nice to group them all together on the one slide, kind of like how they've been doing it along the way. So I decided to include this slide as well. Okay, so Leber hereditary optic neuropathy, so LHON. So what is that? Okay, it's caused by mutations in one of several mitochondria genes. Okay, so it could be any of these. Honestly, I would just kind of like, they all kind of have the same pattern, right? So MT, ND1, ND4, ND4L, or ND6. Okay, so not too bad memorization wise. Anyway, so what kind of symptoms arise with that? You're going to have blurry vision, vision loss. It's going to be a movement disorder of some sort. Also, you're going to have cardiac conduction defects. So once again, between all these different diseases that we're going over in this category, make sure you just kind of know like what makes it different from other ones. All right, now myoclonic epilepsy with ragged red fiber disease, so MRF, okay, caused by mutations in MTTK. Okay. So that one's specific, not too bad at all, right? All right. Now the thing to notice here is that you have twitching muscle spasms, seizures, ataxia. So it's all like muscle slash like movement related symptoms. Thing that really stands out to me is like the muscle spasms and seizures, right? Because that kind of helps you with its name, right? Kind of sticks in your head that way. But yeah, and also that you see these ragged red fibers. Of course, they're not gonna. So on the exam, they probably give you like MERRF. So you'd have to know that what it stands for instead of give you that whole title. So therefore, if they gave you ragged red fibers as like a clue, it wouldn't help you unless you knew what it stands for. So that's something to keep in mind for these questions. But yeah, mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes. So MELAS. Once again, this one's got kind of like several, so it's a little more annoying, but I would probably remember them just in case. I would assume the ones like up front would probably be more common, I would think, but I'd probably just keep them all kind of in my head. Things that are happening, of course, stroke-like episodes. That's the thing that really stands out to me. Maybe the headaches and vomiting. You haven't really seen that with the other ones, but definitely the stroke episodes. That's something. And then like our final one, so Leigh syndrome. So that's going to be ATP6 gene. Symptoms arise in first year of life. That could be something to kind of help you out, because every now and then they'll include like demographic information, like a 1 or 2 year old patient has these symptoms, then that may kind of clue you into this one. So the things that stand out, obviously fatal within first years of life. That definitely stands out. Definitely dysphagia, diarrhea. That kind of stands out to me with this one because we haven't mentioned it yet so far. So just kind of keep the things, like I said, that's what helps you with these diseases is the stuff that's like different from the other ones. That's what always helps you out. And yeah, always know the genes, even though it gets kind of monotonous after a while. Um, but yeah, so I do have, like I said, that exam 7 disease list that I'll put in that Google Drive that I made, that me and Stacy made. So I'll put that in my folder for you guys. I'll also scrounge around during the break and stuff. Well, at least after our exam, because I don't want to spend too much time digging. But of course, if I do just find it on the surface, that Anki that include all the diseases for the semester, I'll share that as well in the Google Drive. So you can go ahead and start studying for cumulative if you want to. But yeah, I think that'll probably do it for us tonight. I'll go ahead and put the thing in here again, the link for the attendance. So I put that back in here just in case somebody didn't do it yet. I really appreciate it, guys. If you do that, because they don't like us to think that we're just talking to a wall. So that's really the reason we do that. I really appreciate you guys coming out on Friday nights all the time. It's pretty awesome. But yeah, I wish you guys all luck. If I don't see you at all, which I probably won't, obviously, because you're in Knoxville for the most part. I wish you all a good Thanksgiving break. Um, yeah, whether you're here, stay in school or whatever. Thanks, appreciate it. Appreciate the good luck. Need it. But yeah, hope you guys have a great Thanksgiving break. You guys deserve it. It's been long, hard journey. But yeah, no problem. No problem.